This is part 6 of the chapter 12 lecture notes, and I apologize for the um, length of this lecture, but it's just a very long chapter. Now, we were talking about multiple alleles, and that should be parasite with an E on the end. But multiple alleles means that there's not just two versions of the trait, but there can be three or more versions of the trait. So, drugs that are used to treat malaria affect the parasite, the Plasmodium falciparum parasite, which is transmitted by mosquito. So, various drugs that affect this parasite um, work depending on whether or not the parasite has drug resistance, has a gene for resistance to the drug. So there are many different alleles, many different versions of the drug resistant gene. And some confer a very high level of drug resistance, and some confer like a medium level, and some confer a very low level of drug resistance. But the point is that there are many versions of the drug resistant gene in the plasmodium. And here I told you that I would look up alkaptonuria. Um, if you Google it, there's a lot more information, but um, one of the symptoms that shows up early on in life is black urine. And this pigmentation that you see here, this is not facial hair, um, is another symptom. Now, we were on this slide and talking about the fact that some traits are linked on chromosome pair number 23. Pair number 23 are called the sex chromosomes. And a male will have an X and a Y, and a female will have an X and an X. And some traits are only inherited on the X, so we call them X-linked. And in Drosophila, which are fruit flies, the gene for eye color is only found on the X chromosome. So a male fruit fly will only inherit one gene for eye color, and that gene can be for brown, cinnabar, sepia, vermilion, white, or red. Now, this example is only showing white and red eye color. It's showing a male with red eyes and a female with white eyes. And I don't know if it's obvious, so capital big W is for red eyes, and little w is for white eyes. White eyes is recessive. So notice that the allele for red eyes is shown here. It's a big X with a big W superscript. And the allele for white eyes is an X with a little w. So the gametes that the female can give are X little w, X little w. The gametes that the male can give are X little w, Y. And as you can see in the offspring, when you work a Punnett square problem, for the female offspring, one is going, or both are going to have red eyes because they're both going to be heterozygous. The father can only give a big W, and the mother can only give a little w. In the males, the male offspring are all going to have white eyes, because ask yourself, where did the male fruit flies get their X chromosome? They got their X chromosome from the mother, and she only had the white eye allele to give. So this shows you another thing about X-linked traits is males inherit X-linked traits from their mother. Males inherit X-linked or sex-linked traits from their mother. Okay, now what we're looking at here is, it says the son of a woman who is a carrier of a recessive X-linked disorder will have a 50% chance of being affected. So if the woman is a carrier, that means she's heterozygous. That means that she has one allele for normal and the other allele for the disorder. So there's a 50% chance that her son will be affected. Her daughter will not be affected, but she will have a 50% chance of being a carrier. Now, I say her daughter will not be affected. Her daughter will not be affected if her husband is not affected. But if the daughter's father is affected, then she can be affected as well. So we're looking at X-linked disorders. Some examples are color blindness and hemophilia and a form of muscular dystrophy. These are examples of X-linked disorders. So if we say color blindness is X-linked and color blindness is little c, so normal vision would be big C and color blindness would be little c. Okay, so if the father is unaffected, then he's going to be X, big C, Y. If the mother is a carrier, she's going to be X, big C, X, little c. They've shown it here on chromosomes, but I thought it would help you to be able to write out the genotypes. So the daughters can either be X big C, in which case they're unaffected, or X big C, X little c, and they're still unaffected, but that tells you that the daughter has a 50% chance of being a carrier. And the son, since the son gets the X from the mom, the son can either be unaffected, X big CY, or the son can be affected, X little c y. All right, some alleles are lethal alleles. So all Manx cats are heterozygous for the capital M, capital L allele, big M, big L allele. If they have big M, which means they're homozygous, they're normal house cats. If they have big M, L, they're heterozygous and they are Manx. If they are big M, L, then they are homozygous for the lethal allele and they will die. So the lethal allele is recessive. If you cross two Manx cats, which means you're crossing two like this, that is a monohybrid cross. So it says you're going to get an offspring ratio of 2 Manx to 1 normal instead of 1 to 2 to 1, because the other one, since in your 4 Punnett squares, one of them is going to be ML, which means lethal. All right, a trihybrid cross. We can analyze, but I will never ask you to do one. But in a trihybrid cross, you're looking at 3 traits at one time. And analyzing a trihybrid cross, you try to analyze each trait separately. So we have 3 yellow and 1 green seed, and I guess, okay, so it doesn't mean you have 3 yellow. It means a 3 to 1 ratio of yellow to green. So for every 4 seeds, for every 4 garden pea seeds, 3 are yellow and 1 is green. And then for all the ones that are yellow, 3 are round and 1 is wrinkled. And for all the ones that are green, 3 are round and 1 is wrinkled. And then for all the ones that are yellow and round, 3 are tall and 1 is dwarf. For all the ones that are yellow and wrinkled, 3 are tall and 1 is dwarf. And you can keep on going with that. But this is a method that's better than the Punnett square method. It's the forked line method, and it's better when you're looking at 3 traits at one time. Um, and it shows you here the probabilities out of a total of, you'd have to add these numbers up, 27 plus 9 plus 9 plus 3 plus 9 plus 3 plus 3 plus 1. So what is that? We know 9, 3, 3, and 1 is 16. So that's 16, 17, 18, 19, and 19 plus 18. I should have done this before. It's 37, and 37 and 27 is 64. So all of these are out of 64. If you wanted to show it as a fraction, it would be, if you wanted to tell how many would be yellow, round, and tall, that means they have all the dominant traits, then it's 27 out of 64. All right, now there's another thing that happens is crossing over, which we have talked about already, and this is what crossing over looks like. Crossing over means that instead of just having these 3 genes and these 3 genes, either big A, big B, big C, or little a, little b, little c in the gamete, you can have these other combinations as well due to crossing over. Okay? So crossing over again and independent assortment are the 2 ways that you get um, genetic variation. Now, another type of inheritance is called epistasis. Epistasis goes like this. The mottled agouti color A, big A, is dominant to solid coloration, such as black or gray. A gene at a separate locus, C, is responsible for pigment production. The recessive little c allele does not produce pigment. So a mouse with the homozygous recessive genotype, little c, is going to be albino regardless of what's present at the A on the chromosome. So this, what the epistasis is the C gene is epistatic to the A gene. So what you can have is you can have 2 big A's and 2 big C's, and then that's going to be um, the mottled agouti color. It looks brown to me, but it's a mottled color, evidently. Or you could have 2 little A's and 2 big C's, and you would get a black mouse. And then you could have 2 big A's and 2 little C's, and because you have 2 little C's, you're going to have albino. Albino results no matter what the A's are. So, so this is just a Punnett square showing, and it would have to be a dihybrid cross, but it's showing the possibilities you would get if you had 2 mottled agouti colored mice that were heterozygous for the A and the C. So the dihybrid cross would give you 9 out of 16 would show the agouti color, 3 out of 16 would show the black color, and then 4 out of 16 would show the albino color because all those 4 are going to have little c, little c. So we've got enough time. So for the types of inheritance patterns that we have looked at simple dominance, we have looked at incomplete dominance, we have looked at epistasis, and we have looked at multiple alleles. These are the inheritance patterns that we have studied. We have also studied X-linked traits. I'm sorry. So make sure that you understand those, each of those inheritance patterns. Simple dominance, incomplete dominance, multiple alleles, and X-linked traits.